Okay, lecture 13 we had some students miss out on this lecture, so I'm redoing it, posting it to Blackboard, and we'll give them an opportunity to get caught up. So we did that lab that day. I'll have to check with the students to try to make up those labs as we move forward. So one of the things we hope we're going to get to in some way, shape, or form, teaching you how to singe udders. It's a lot easier than you think. If you follow a couple simple principles, it's fairly safe and fairly easy. Most of the time, the animal, if you do it right, the animal shouldn't even know you're there. So there's also an alternate method we'll demonstrate from a former student. He picked it up at a internship. You can use a spray gun that forms a cone spray, 70% alcohol. You blow it over a lighter or another flame source, and it will help reduce the hair on the udder. So the purpose of singeing or clipping udders is to lower the bacteria counts and reduce the prep time on your cows. You're looking for cleaner udders. We're trying to keep the udders clean and the milk clean with this. So if you compare the socks before and after, we did milking before and after. There's a lot less debris as we bring up those teacups on our milking machine. If there's hair there, there's usually a sawdust stuck to it that can get sucked into the milking machine. The socks can collect that, and if there's bacteria on there, you've got warm milk going through the socks. Those bacteria are going to grow and multiply and end up in the milk. So pretty obvious on this one. Not quite as obvious here in this picture, but there is a definite difference if you look up the sides, if you look across. We also have to do a better job checking for mastitis. So when we're thinking about bad microbes, we need to remember, trying to keep the bacteria out of the milk, what those bad microbes need to grow. They need to be present. They need a food source. They need a source of water. They need air. They need warmth. And they need environment free of irritants and inhibitors. And when we're thinking about mastitis, we have to think about the challenge versus the defense. So, how much challenge there is, the more bacteria we get on a teat end, the more likely she is to become infected. The better our defense is, how we clean, how we keep things clean and dry, the less chance of bacteria getting to that teat end and causing problem. So, from an udder prep standpoint, we're thinking about lowering the bacteria on the cow, and hopefully that will lead to lower bacteria in the milk. So, historically, we've got to keep those animals clipped, keep the bedding underneath them clean. We don't want manure caking up on these animals, and therefore being on the udder and possibly introduced into the milk. To paraphrase the way I was taught, management is the barrier between the bacteria that are all around the cow and the inside of that udder. So graphically, will look like this. Management is that shield in the middle there, trying to keep the bacteria from getting into the teat end. So how do we do that? We want to make sure we don't spread milk from cow to cow. Whatever bacteria is in one cow could possibly end up in another cow. So we want to make sure we don't spread that milk from cow to cow. If we're using some sort of common rag, some sort of common cloth, we're going to take the bacteria from one animal to the other, and we don't want to do that. Milking management, the goal has to be to keep the bacteria out of the milk. wherever the milk is. So the milk can be in the cow, so we don't want the udders to become infected. The milk could be in the milking machine. We want to make sure our machines are clean and we're not introducing stuff into the milking machines as we go through. And then ultimately, that milk ends up in the tank. We want to make sure that we do not introduce bacteria into that tank and allow them to grow and multiply. So we want to make sure that we don't contaminate the udder during milking. So we want to make sure the udder is clean and we keep it clean. And we want to make sure that when we're cleaning stuff up, if we've got hoses, if we got bedding, we want to make sure that that material does not get splashed back up on the cow. So good practice is not to use hoses while the cows are in the parlor. Make sure you scrape down your stalls before you start milking if you're in a tie stall situation. Back in the old days, we used to wash the entire udder, and then once we washed it, we'd attempt to dry it. But one of the downsides of all that is where if the udder is still pretty hairy, it's pretty difficult to dry down completely. We know water left on the udder will follow gravity sticking to the skin and move down. Usually the low points are the teats. And if the cow is being milked, that water is going to collect on top of the liner. So the Langston picture you see here, water is collecting between the teat and the liner. If there's a liner slip, that cow tea, all that water that is soaked up on the cow will end up in the milk, and we'd really not like that to happen. So in recent years, last 15, 20 years, we've gone away from use of water in the parlor, use of water in milk prep. So we tend to just pre-dip, and then hopefully the udders are clean on the animals coming in. We pre-dip, wipe that off, and we attach the milker. We have a minimum amount of water involved, so hopefully that minimizes the amount of bacteria that end up in the milk. So if we're thinking about post-dipping, what are we trying to do here? The middle and end portions of the teats are dilated for up to 2 hours after milking. The teat canal might remain dilated for longer than that, slightly dilated. So if we put a dip on after milking, we put a barrier there, especially while it's open. So if any bacteria do get there, they're killed before they're pushed up into the canal. So teat dip is attempting to kill the bacteria on the teat end and not allowing them to live and enter the teat canal into the udder. So we want to dip as soon as possible after machine removal. We want to do that as quickly as possible, as soon as we're done milking, to make sure that that opening, that dilated opening, is defended. So we're looking at half to 3/4 if you're doing some sort of dip. If you've got scrubbing like we do up at the part of the bubble things, cover the whole teat. And then we form that droplet down on the base there. If you're looking at that, this droplet down here is a good barrier for what's coming through there. Also, if you go up high enough, you're going to have teat dip slowly working its way down, and that's going to continue to flush this area and defend that teat end over time. The milking routine, we're looking for something that's the same all the time. This is important for milk letdown, but also just the cows like consistency. So everybody has got to milk the same way. Everybody doing it their own way is not going to be productive. We've seen in experiments where consistency has been shown to increase milk production 5.5% over not doing it. So we need to make sure that the cow knows what to expect and what she expects happens. There's no surprises, no jolts, no what the hell you doing down there from the cow that potentially leads to an epinephrine or stress response. So advanced milking procedures, after pre-dip, we're going to force strip possibly. We're going to use individual wipes on most farms, 30-second contact time or 45 depending on the label. We've got no water, so we're going to use gentleness. We're going to make sure we're milking clean, dry teats. And we're going to monitor our first contact with that animal to machine attachment. We want to make sure that time frame is consistent over time. We want full milk flow when that machine is attached. We don't want to get there too early. We don't want to get there too late. We don't want to be milking empty cisterns, the teat cistern, the gland cistern. And we want not to be there late where the whole system backs up and we kind of waste that oxytocin, that milk letdown reflex. We want to make sure that we think about how we wipe first. So when we're cleaning our udder, we want to work far away back towards us. And this has to do with the arms. Your arms can contaminate those teats. Most of us take a look at our arms during milking. Things are pretty dirty. So we want to start far away and work back and then attach things in such a way that we don't infect things. So we're looking at milk quality. Somatic cell count, cells of the body have to be less than 750,000 in the USA. There's a lot of places that are taking much lower or cutting you off if you're any higher than 400,000. This is an attempt to cut down the surplus at the plants and also making it legal for export. Most countries will not accept over 400,000 somatic cells per milliliter. If you think about it, how small a milliliter is and how many cells we're talking about, it's probably a good idea. So somatic cell count is the white blood cells predominantly in the milk that originate from the cow. So if there's an infection there, a bacterial invasion, the body responds by sending white blood cells there to destroy those invaders. Somatic cell, meaning of the body, those cells may also come from the sloughing off of the inside of the udder, the secretory tissue falling off the cow. That secretory tissue tends to fall off a lot easier in older cows, so as a rule, higher somatic cells tend to be associated with older animals. So somatic cell count, we have to remember, is not a direct measurement of bacteria. It's an indirect measurement, and high somatic cells, so looking at the millions level, is associated with udder inflammation because those white blood cells bust through the secretory tissue. They're going to lead to lower milk yield and quality as those secretory cells are pushed off and the blood starts to leak into the milk. That's not something we do, so the milk yield and quality will go down. So somatic cells are associated with the cells of the cow. What we're really concerned about is the bacteria in the milk, and there are a number of different measurements that come back from the plant. So here are 4 of them. We have the standard plate count, is a measure of the total bacteria present in the raw milk. So we take the milk, we plate it out in a petri dish, and we count the colony forming units. And I think it's 24 hours. So that can be called standard plate count or plate count. Our PI count, preliminary incubation count, is the estimate of cold-loving bacteria. Laboratory pasteurization count is the estimate of bacteria that can survive pasteurization. Coliform count is the estimate of bacteria that are from the manure or contaminated environment. So standard plate count generally gets all of them. The PI count, the lab pasteurization, the coliform count are all subsets of the standard plate count. So if you have a high standard plate count, you got to start thinking about, okay, where are these bacteria coming from? Are they coming from the cow? Are they coming from the equipment? Or are they being introduced at the tank level in some way, shape, or form? So these are the links to the handouts that I believe everybody now has copies of. I've handed you those. If you need more, that's where to get them. So we'll talk about these as we move forward. So you've got D-Lel Val 12 golden rules of conventional milking. So these are updated. They just used to do one type of milk. So before milking, the 4 things we want to consider is monitor udder health regularly. So checking for mastitis, checking for bacteria, checking for somatic cell, milking the right order. You want to milk your healthy animals away from your unhealthy animals. If there's some way we can do our mastitis cows last and do our young fresh heifers first, um, the hope is that you'll keep the milk high quality, low chance of antibiotics, low chance of bacteria. They are suggesting that you always fore milk, so that provides for a good stimulation of milk letdown and also an opportunity to check for flakes and milk that doesn't look right. So clean and disinfect teats before milking. Again, clean, dry teats are what we're trying to milk. The 4 that they list for during milking, and again, there's a lot more details on those handouts. You got to check your milk vacuum. So that's up high in a parlor up at the dairy. So checking that every once in a while, make sure that's where it's supposed to be. You want to attach the cluster, the milking unit at the right time. So in that 1 to 2 minutes after that first stimulation in their situation after you fore milk. We want to avoid over milking, so get those machines off as the animal stops secreting milk or letting milk go. And then we want to detach the cluster correctly. We want to shut off the vacuum, let the machine fall off. We don't want to pull that machine off under vacuum because that can lead to tissue damage as well as an increased opportunity for mastitis bacterial infection. After milking, we want to disinfect those teats immediately, so some sort of post-dip. We want to clean and disinfect our milking equipment to get it ready for the next milking. We want to ensure proper milk cooling. The faster we can lower the temperature, the quicker we take one of those things away from our bad bacteria. We take the warmth away, and they'll grow less fast, keeping the bacteria counts low. Don't give them a chance to multiply. Then we need to go through and make sure we monitor our milking, check things, maintain things as we go through, check to see if we're getting good timing, we're getting good milk letdown and catching that, attaching the machine at the right time. So there are factors we'll talk about to make sure you can evaluate what you're doing right. Automatic milking, so robots. So the same sorts of things here. You're going to monitor that udder health. The computer is going to help you identify the animals with a temperature, identify the animals with high somatic cell or high electrolytes in the milk. Make sure that your milking interval is good. The animals that aren't going up to be milked, find out why. Go check on those animals. Check your quarters. Make sure everything's where it's supposed to be. And ensure that the machine is doing the proper prep. That prep should create a good letdown. It's rather vigorous, at least the ones I've seen. So you're going to do the same thing again with your during milking. We're going to check the vacuum. You're going to check to make sure the machine's working milking attachment. You're going to avoid over and under milking by setting your limits. When it drops below a certain amount, that teat cup comes off. And then you're going to check, make sure that everything is set the way it's supposed to be. Again, post milking, we're going to do what we can to prevent post milking infection, some sort of dip. If we can keep those animals standing, that's a good thing. We're going to make sure that the system gets clean between cows, the milk gets cooled promptly, and we're going to monitor the milking and feeding reports associated with that computer unit. So moving on to lecture 14. So lecture 14, we had those take-homes due then. I think everybody's turned those in. Hopefully we'll have some tests later on. We can think about different things we might be able to do to lower the bacteria on the cow. And hopefully this lowers the bacteria in the milk, keeps our milk quality up. So if we divide them kind of into different categories, it might look something like this. If we consider which ones are equipment related, you've got the maintain your equipment, checking for defects, checking for cleanliness, checking for leaks, breaks, all that stuff. And then cleaning our equipment, making sure we put it through the right concentrations of chemicals, make sure we put it through the correct temperatures, make sure we do all we need to do. Which animal management other than milking time, we need to clip and or singe the udders. Singeing udders a little bit safer than clipping, a little bit quicker than clipping. So that's why it's used often. We want to keep the animals out of the mud. We want to keep the stalls clean and use the partial insertion of treatment tubes to promote not pushing bacteria up into the teat canal, in the teat cistern. And then during milking, all the things we might do to keep the bacteria to a minimum on the cow. So individual wipes. We're not going to be transferring bacteria from one cow to the next. We're going to make sure we have an approved teat dip. We're going to make sure that dip is clean, so there's not a lot of manure and sawdust and all that stuff in your dip cup. Make sure your hands are clean or covered with gloves. We want to make sure we don't get milk from cow to cow. That might be within the little rags. That might be with your hands. That might be with the machines. So we want to make sure that the milk doesn't get from cow to cow. We're going to clean the equipment as we go through if we can. Not too much water, but if we have to clean things off, we will. Do not contaminate the udder during milking. So making sure that we don't splash water around or let things fall down in some way, shape, or form. No water on the udder, and we want to post dip. So all these should help us lower the bacteria on the cow and hopefully lower the bacteria ultimately in the milk, lower our chance for mastitis. So what do we need to do out in our facilities to maintain our top quality milk? So barn, we're going back to clean and dry. Everything needs to be clean and dry. The less bacteria we have on the cow, the less mastitis we get, the less bacteria we get in the milk, either going from the cow to the milk or from the mastitis to the milk. We're trying to keep the bacteria down in some way, shape, or form. We got to make sure our milking equipment is maintained and cleaned. And then when we are in the parlor, we're trying not to spray stuff down, move bacteria all over the place when cows are there. So bacteria and somatic cell are certainly affect milk quality, but there are other things that we might consider. There are other bacteria counts we need to be mindful of, but those are usually special cases. There are certain things that will lead to off flavors. So if a cow eats something if it's fat soluble, it'll end up in the milk. Those off flavors may lead to milk that's kind of, well, it's not quite right. So the Wagner Farm, Juvendale over in Kaz, they just had their auction and sold out. Vince and Julie were big proponents that you do not run the vacuum cleaner, or vacuum cleaner, the barn cleaner, while you're milking. They said that stirs up the ammonia and smells in the barn. Any air that goes into the milking machine, any vacuum that's generated, will be filtered through the milk, and those milk will absorb those flavors. So I'm not sure that's totally correct, but good ventilation, making sure, and they had fantastic ventilation there, will definitely keep the animal from breathing in those smells and those smells ending up in the milk. I think it's a good idea with not cleaning the barn while you're doing that, tie stall, but I'm not sure what percentage of the problems. Another way to discuss milk quality would be components. How much fat I have, how much protein I have. That could be a way or another way to look at milk quality. So here's an article that drives home the point about animals eating. So this article is up on Blackboard if you want to read more. But this was a study done about the Parmesan cheese industry in Italy. And they tried doing an American approach, TMR and things like that, to feeding their cows so they get more milk and therefore more cheese ultimately. But the problem was the cheese didn't taste the same as it would. And it really comes back to, you got pasture animals and TMR animals. Those pasture animals were eating compounds, eating plants that led to a unique flavors of that cheese. So the real Parmesan that's actually made in Italy, flavor of the Gusana cheese, they found was affected by what the cows ate. So this study was presented in 2006. Here's the pasture composition, kilograms or grams per kilogram of dry matter. So if you're going to look at it on a percentage basis, you're going to back off the means by 1 decimal place. So if we look at the top here, dry matter grams per kilogram, 244. That means the dry matter percent was 24.4%. Crude protein 15.2, NDF 40.7, AD lignin 5.7, digestibility 65.5%. So you've got that there. So diets were pretty similar, pretty typical what you expect for a dairy cow, especially on pasture. They took these samples and they generated steam. So the steam over here on the left, they boiled it through. The compounds got boiled off. They went through a condenser, and then the stuff that came off was analyzed. So all the smells and tastes were lifted out in the steam, and they were able to quantify them. So here's the compounds they found in the pasture-derived cheeses, and then the estimate or guess of where those flavors came from. So you have peach, orange, green hay, rose, vanilla, daisy, herbaceous, green flower, sweet, and fresh flower. Most of these at least have a semi-positive connotation to them. These are the flavors in the cheeses. They found them in most of the feed samples that they identified. So the peach flavors were found in all 13 species on the pasture. Green hay was only found in 12 of 13. Sweet was only found in 2 of 3. going through. So they're showing that the flavors that were in the plants or odors that were in the plants were ending up in the cheeses. Here's the compounds or flavors or odors that were found in the pasture cheeses, but they couldn't find out which plants they came from. So you have jasmine, nutty, onion, fried, lily, buttery, hot milk, the compounds, and then we're looking at they probably came from an overlooked species. Some cows are eating things because they follow the cows around, watched what they ate, took samples. Maybe they didn't see where the jasmine came from or the nutty came from, um, or they're figuring that something was taken into the body, metabolized, or generated, and that somehow ended up in the milk. And those are flavors there. Again, they're not what you'd call objectionable. What they did is then compare it to the TMR derived cheeses. So they had the things that were in those cheeses in addition to all the other flavors, but not in the pasture ones. So we have sweaty, woody, body odor, and metallic that are coming from the TMR derived cheeses, but not the pasture cheeses. So you're assuming that these came from, on some level, the TMR. So sweaty, woody, body odor, metallic, those are things that we're probably not going to like, but this is the reason that the cheese did not work or taste right with the TMR derived cheeses. So milk quality, lots of different ways to think about it. So, getting back to how do we prep our animals, particularly if we're going to treat them, dry cow treat them, something that we've got injections for all 4 quarters. So scrubbing sequence, we're going to start farthest away and work back toward us. The way this picture looks, I'm more inclined to do with the numbers there, 1, 4, 2, 3. But I think it's meant to be more you're on the side, you're working from the far side of the animal back towards yourself. I'm looking at more of a diagonal on these animals. And so the idea behind that, as you scrub those teat ends with an alcohol swab, you're moving your hands away from those so you don't touch those teat ends anymore. So they can hang there in the air. They should remain sterile as long as you don't touch them, at least for a short period of time. And then if we're going to go back and treat, like say dry off tubes or something like that, we're going to start with the closest to us. So we can go and we do that tube, we do that insertion. So the first thing that touches that is the sterile end of that treatment tube. And we should dip that after, but we're starting closest to us and working away. So we don't touch that teat end with our hands or arms and then take the treatment tube and push that bacteria up into the udder. So start sanitizing coming towards you. You can do that for udder prep as well. Treat or use those tubes going away from you, and make sure you dip after everything's done. So you can still do the full insertion with tubes, but more often than not, we're doing the partial insertion, which is what's here on the right side of the frame here. The idea is that we're putting that tip in far enough to get whatever material we want to inject, whether that's a barrier, whether that's antibiotics, up into the teat cistern, ultimately the gland cistern, but we're not going through and pushing out the keratin plug. If we do the tip on the left, we're going to push out that keratin plug, and it's going to take a day or two for that to reform. That keratin plug is bacteriostatic, so it doesn't kill the bacteria, but it stops them from growing, which is very important. So the more we can leave with the tip on the right side, the better off we are. So, talking about the milk ejection reflex. So, why doesn't milk just run out of the teats? And well, you're going to say, well, it just does. You know, I've seen cows leak all over the place. Well, that's not what I'm referring to. What I'm referring to is why doesn't all the milk come out? Why doesn't it fall to the bottom of the udder? And that's based on capillary action and vacuum. So if we take a straw, we have a glass of milk, we dip the straw in the milk, we put our finger over the end of the straw, we lift up that straw, it's going to take what milk or whatever liquid we have into that straw. The only way to let that out is to um, take your finger off the end, the air comes in the top of the straw, gravity takes everything going down. So as long as I have my finger on the top of that straw, I have a vacuum. The vacuum is going to hold up the weight of a certain amount of liquid. It's not going to, and a certain diameter of liquid as well. The milk wants to stay together because it's a liquid. It's going to try to stick to the wall, stick to itself, and form that barrier. So the only way I can get it to run out is to put air in behind it, and then it drops down. If we look at an alveolus moving through, you're going to have the same sort of thing as a straw with your finger on the top of it. So you've got all this milk being formed in the lumen of our alveolus up top, but there's no air inlet in there. The water wants to stick together, the water wants to stick to the side. The only way to get rid of it is to squeeze it out. We can't let air in and let gravity do all the work. We have to squeeze the top of the udder, all the secretory tissue, to move all that milk down to the bottom of the udder. So it's all dead ends. We don't have a thing, we can't lift our finger off the top. If each alveolus had a little air hole in the bottom that could be open and closed, we could just open that and gravity would do the work. But because we don't have that, we have to have the milk ejection reflex. So, what is the milk ejection reflex? What are the two parts? What are the two phases of that? So, looking at our diagram here, we have the stimulation of the udder. We're cleaning off that udder, getting ready to go. A calf nuzzling the udder, trying to find that teat end to suck the milk out. So the first part of our reflex is the nerve impulse. That's going to go in milliseconds. So snap of a finger. She knows what's going on. That signal goes up the nerve endings into the spinal cord, into the brain. The cow realizes what's going on. It takes all those signals, it aggregates them together and sends a signal to the hypothalamus. The hypothalamus at the bottom of the brain's going to send a signal to the posterior pituitary to let the oxytocin go from the posterior pituitary. That oxytocin is going to flow from into the bloodstream, the superior vena cava to the heart. It's going to flip out through the lungs, back to the heart, and then out the aorta to the lower half of the body, and eventually to the udder. So that's the part that takes the time, the flowing through the bloodstream. So we say milk letdown takes 1 to 2 minutes. That's because of the flowing or the transport of the oxytocin from the brain to the udder. Once we get to the udder, the oxytocin somehow connects with the myoepithelial cells. You see these little eyes there with the tendrils going out in each direction. Those muscle cells wrap around the alveolus. When oxytocin gets there, that triggers those to shorten. As they shorten, they're going to collapse the alveolus and push the milk, squeeze the milk like squeezing out of a sponge to the lower part of the duct system, the teat cistern, the gland cistern. So squeezing all that on the top, just like you can leave a sponge on the counter. They're still holding water. You pick it up, you squeeze it, you're going to squeeze stuff out. Our goal, and here we're moving into lecture 14, we want to initiate a strong milk letdown, and that's going to lead to efficient and complete milk harvest. So we need a strong oxytocin surge, and we need all that oxytocin to get to the udder. And then we need to attach the machine. So we get the strong milk letdown, we attach the machine in such a time that we catch that milk as it comes down. We aren't there too early, we aren't there too late to lead to a backup of milk in the system and a need for a second milk letdown. When do we attach the unit? We're hopefully going to go somewhere between 1 and 2 minutes. And it really comes down to, in your milking prep routine, what is your first stimulation? Is it when you get to the cow? Is when you turn on the milk pump? Is it when you wipe those teats off? Is when you dip those teats? At the dairy, it's about wiping the teat ends off. That is thought to be the vigorous with the towels, the vigorous stimulation. We're going to get that in some way, shape, or form to lead to a good milk, good oxytocin release, which will lead to a good letdown 2 minutes later as it pushes the milk from up in the secretory system down into the duct system down below. So to make sure we get that milk ejection reflex, we need a strong one. We need to be patient and consistent with our cows. We don't want to rush them. We want to do things on a cow time, a cow schedule, and we need to do the same things over and over again. As we talked earlier, consistency leads to more milk production. Anytime a cow goes, whoa, 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 what's going on? We're going to get a slight stress response, and that stress response is going to cause problems with our milk ejection, milk letdown, and harvest. So we're looking for sameness. Every day is Thursday to a cow. We want every day to be the same, every milking to be the same, no surprises. We want everything to work.